Hello and welcome to Science Unscripted. It's Connor here.
And Gabe.
Today we are going to be talking about gain-of-function research, or GOF research, the idea that you take a virus or a bacteria or any kind of pathogen and enhance it somehow artificially in a lab. You can make it more deadly, you can make it more infectious, you can make it bioluminescent, you can do all sorts of things. And that's a fact right now that researchers around the world are doing that in labs. They're doing gain-of-function research where they take dangerous stuff and tweak it.
And we're gonna be talking about what it is, what it's for, because it's not dangerous. Because if these things get out of the lab, like you said before, they can be more deadly. They can spread easier. It's like taking a virus that already exists and making it even worse. What happens if something like that were to happen?
Right.
Could possibly cause a pandemic. So we're going to be speaking to someone, an expert, who herself doesn't do gain-of-function research, but is definitely in the know. And we're going to spend this entire show talking about this stuff because, yeah, if it is dangerous, what's the point of doing it?
Well, this all comes on the backdrop of a COVID-19 debate, wild speculation. We'll talk about that again later, about where SARS-CoV-2 actually came from. And it's led to people focusing on this kind of research, whether for good reasons or bad reasons, whatever. And whether or not we should be doing it in the first place in the context of a lot of lab leaks around the world, not with, super viruses or genetically engineered viruses, but with other viruses.
Yeah, you've got some papers there in front of you full of examples of lab leaks.
Every couple of years or a couple times a year, lab leaks. And again, not enhanced viruses, but other viruses getting out. Anything from anthrax to smallpox to polio to Marburg, you name it, Ebola. It's been leaked from a lab. Yeah. And if we're toying with other viruses and making them super viruses, could that not also happen? And what would be the consequences? Let's
hear about that from Silke Stertz. She's from the University of Zurich, out of the virology lab there?
Medical virology, and she has no vested interest in keeping this research going. That's important. She's not part of a team doing it now. She's in the field of influenza. And yeah, that was important to us to talk to her about what gain-of-function is actually accomplishing and whether we should be doing it at all.
Science Unscripted.
My name is Silke Stertz. I'm a virologist focusing on influenza viruses. And as an influenza virus researcher, I've followed the gain-of-function debate over years. But myself, I have not been involved in any gain-of-function experiments.
Can you briefly explain what is gain-of-function research?
So gain-of-function is probably not the right term for the discussion we want to have. Gain-of-function just means you give a virus a function that it didn't have before. And most experiments that fall under this are not problematic. We can, for example, label a virus so that we can then see which cells get infected or where the virus localizes. What people are worried about is gain-of-function of concern experiments. Those are different.
What does that mean? What are those harmful manifestations of gain-of-function research?
It's if we give the virus a function that is of concern, meaning that we make it, for example, resistant to current treatment options, or we give it the ability to spread and transmit better, things like that are of concern, that make the virus worse or more dangerous. Those are the experiments that are under debate.
So the picture I have when you talk like that is a laboratory where researchers are making viruses more powerful, they can spread more easily, or more pathogenic, they can kill more easily. Why would they be doing that?
We want to understand how viruses cause disease and what makes a virus transmissible. If, for example, you think about influenza viruses, we've heard about the bird flu, H5 viruses, for a long time. And people are very worried that these viruses might make the jump and cause a pandemic like we saw with SARS-CoV-2. But in order to see if this is happening, if we need to prepare ourselves, we need to understand what makes a virus transmissible. And to answer that question, one option can be to make a virus transmissible in the lab.
Before we get into the ethics or into the point of this research, how exactly do you do this? How do you change a virus or amend it or add something to it?
We have genetics methodology to modulate a viral genome. Their genomes are very small, much, much smaller than, for example, the human genome. And for these small genomes, we have tools available to modify them. We can introduce mutations, we can introduce foreign DNA or RNA, and we can also take things out. These methods have been established for quite a while and helps us understand how viruses work.
Silke, one of the most famous or infamous gain-of-function research experiments is in your field, influenza. And this was Ron Fouchier,
Yes.
if I'm pronouncing his name right, making the H5N1 virus extremely transmissible and pathogenic and dangerous, really, really dangerous. And it caused a lot of controversy. I'm wondering how you view that. How do you view his experiments?
In my opinion, these experiments were absolutely important and helped us understand a lot more what makes an influenza virus transmissible. He has highly trained personnel and very high security labs. So in my opinion, these experiments could be done in a biosafe manner. And I think we've learned a lot about them. It's clear that such experiments that are probably at the upper end of what we can and could do, that they need a lot of approval and a lot of discussion beforehand if this is the right thing to do. But in my opinion, these experiments were justified.
And how would you explain that to someone who's not in the field? Let's say for someone, let's say you told someone on the street, there's this Dutch scientist fooling around with viruses, and he has made this bird flu virus, possibly dangerous for the entire world. If that virus had leaked out of the lab, it could have caused a pandemic. How would you explain to someone like that why this research is good or justifiable?
Well, we have seen with the SARS-CoV-2 pandemic that, zoonotic transmission of viruses and pandemics can happen from a natural origin. Viruses can change their host and adapt to their new host, and it is possible that we have a pandemic not only with SARS-CoV-2, but we've also seen that with influenza many times. And it is important that we understand how the viruses do that and which viruses can actually make the jump into humans. And if we understand that, then we have options to monitor the viruses that are around and to focus our efforts in preventing the next pandemic. This is not going to be done with one experiment done by Fouchier, but we need to increase our understanding of what makes a virus transmissible. Then we can counteract this.
One of the critiques that you hear with regard to gain-of-function research is that, well, we just had a pandemic. And the gain-of-function research that, presumably was happening before the pandemic didn't help us in any way, shape, or form to stop this pandemic. We basically had to take the virus that we discovered because of zoonotic transmission and analyze it, sequence it, and then find a vaccine based on that and not based on some, creation in the lab. How would you answer that? Is this research leading to actual things that help us for the next pandemic?
Yes. The research before the SARS-CoV-2 pandemic has largely focused on influenza viruses. And these viruses are still a large zoonotic threat, and we need to keep studying them. But we have learned now that we have not paid enough attention to the coronaviruses, that they have this large pandemic potential as well. That is something that we had overlooked. And so you can't, from a flu study necessarily then say you have also gained knowledge on coronaviruses. We have to broaden our efforts to also study these coronaviruses. But I think it's also not true that we haven't learned anything that we could use for the pandemic. There has been coronavirus research before, also in the highly pathogenic ones like SARS-CoV-1. And, efforts from these studies developing experimental vaccines in the lab has also helped with developing vaccines and treatments now during the pandemic. But it's basic research. I think a fundamental aspect of that is that you can't always predict what exactly you learn, but we increase our understanding, and that gives us new possibilities.
Who exactly in the world right now is doing gain-of-function research? Because when we were looking around here in Germany for interview partners, we wanted to talk to someone doing gain-of-function research. I couldn't find anyone.
Yeah.
Are they all hiding in their labs, hiding in their caves because this has become such a controversy, or are there so few scientists doing it?
There are really few labs that can do that because you need very high biosafety environments, really state-of-the-art high containment labs, and these are expensive to build, expensive to maintain. You need to train the personnel. So it's really limited in a sense to certain labs. And the other point is that also this whole discussion or debate has made it difficult and people stepping back from it. So I think in Europe it's the Fouchier lab in Rotterdam, but as far as I understand, they have also not been able to continue with that line of work, if I'm correct.
This is going to show some of my biases or prejudices, but I kind of assume the Dutch do this well, properly, correctly, with biosafety measures in place the way they should be. I kind of assume that Swiss authorities do the same and that German authorities would also do the same. There are a lot of countries around the world doing this research, and do you think countries are doing this properly all around the world? Is there an all-encompassing, global rule for how this has to be done or can't be done, or is it up to each country doing it on their own?
It's regulated on a national level. There are no binding rules for all the countries. However, it's not that a country that doesn't have the resources or the infrastructure will all of a sudden start this kind of work. It's very expensive to build these types of labs. And so it's straight away limited to country that have the resources. It's not that you have these experiments going on everywhere in the world.
Yeah, so we're going to take a short break from Silke Stertz at the Institute of Medical Virology in Zurich, Switzerland, to go to the streets of Bonn, Germany.
Beautiful cobblestone path around just right next to one of the very many fountains that you find in Bonn city center, and the kind of thing that makes me really happy to be in Europe, in a city in Europe where you can just go and listen to this music. It's everywhere. This is a guy riffing on a clarinet on the Godfather. That you didn't hear that part before, but that's just him taking those chords and just riffing.
We were there walking around because we wanted to get back to the, you could say, the origins of what this whole interview is actually about. Why is anyone talking about gain-of-function research? Almost no one ever talks about it, but it's a discussion point in connection with the pandemic.
Yeah.
Let us be clear.
Yeah.
No one knows where SARS-CoV-2 came from. No one knows.
Regardless of what you think or anyone else thinks or what you've read on the internet, there is no evidence that the SARS-CoV-2 virus came out of a lab.
Right. And there's evidence that one of the initial outbreaks of SARS-CoV-2 was at a seafood market in Wuhan, in a city in China. But that is not the same as saying that's where it came from. That's not the same as saying that was the origin. The origin is still up for debate. And when I say nobody knows, I should be more accurate. We don't know. It's possible someone out there knows. I don't know. You don't know. And the internet is full of speculation. But we still thought it would be interesting to go to a different market, a market that also, frankly, sells fresh meat, a big butcher's stand, cheese stands.
And fresh fruit.
Fresh fruit, yeah. In Bonn, Germany, to just ask regular people there, where do they think SARS-CoV-2 came from? Where did the COVID-19 pandemic originate?
I think it came from our stupidness, worldwide stupidness.
Can you elaborate on that?
Oh, in English. It's said it came from China, from the bats. What I think about it, I don't know. I don't think that science already have an answer, as long as I followed the discussion. Of course, I read a lot of articles in the beginning, but then my interest vanished because the focus was on living with COVID then and not how it came up.
So you believe that the COVID-19 virus was made in a lab?
Yes.
What is your evidence for that? Or why do you believe that?
Maybe it is from my stomach. Yeah, this is my feeling. I cannot 100%, but I see the behaviors, the plan behind, and how they play with us, with the people. I would say this is my feeling.
I personally have no idea, so not any suspicions.
When you say you have no idea, does that mean you've never heard anything about where it came
I did
from?
about theories about China or something, like in labs or I don't know, but I think that's all a bit over it.
So you think it came from the bats then, or what?
Maybe. There are so many options, there are so many viruses, where did they come from?
But is it fair to say that you basically don't care? This is not a topic that interests you at all?
It is there, so why do we need to search from where it come,
Okay, let's turn from the couple of people on the streets of Bonn, not exactly scientific, to...
No, yeah.
What do you got there?
A glossy
COVID-19 origins?
report from the National Intelligence Council in the U.S. It's very readable, probably about a dozen pages total, but I'm going to flip through the bullet points.
On their conclusions on the origins?
Yeah, just so all our listeners out there can talk about this reasonably. And there are basically 4 possibilities for what this virus is or
All right,
was.
rattle them off.
Okay, number 1. Was it a biological weapon? The idea that a country would be developing it in secret to use as a weapon? Probably not, according to this report. I'm not going to go into the details as to why, but that one you can shove aside as a conspiracy theory at this point. It really probably was not that one. And then most analysts assess that SARS-CoV-2 was not genetically engineered. So that actually goes against this whole interview we've been doing the whole time. This again, or not against it. It's not a gain-of-function virus as far as analysts can tell.
At this point, there is no evidence to make that conclusion.
When they look at it, there's no site they can look at and say, oh, humans messed with that.
Yeah.
It doesn't look like that.
That's not the way it looks like in nature. It must have been amended genetically.
They cannot say that. Even at the cleavage site, that was one of the points that people zoomed in on. No.
Yeah.
So what are the 2 plausible origins at this point? Well, we've discussed them. There's the seafood market.
Crossover.
And they say, look, that could have happened. You have all sorts of people going there and all of these mammals moving around.
Raccoon dogs, bats.
Yeah, and that's very plausible in the sense of it starts spreading, no one's sure where it's from, and it was a cluster hot point. And then the idea that it was an inadvertent lab leak, that's one we kind of talked about earlier in the show, that at the Wuhan Institute of Virology, that they would have been collecting viruses, coronaviruses from bats from all over China and bringing them there, and that somebody may have gotten infected, and then you had community spread. Also a possibility, inconclusive, lack of evidence. They don't know. And I just want to make sure that everyone knows that those are the 4 options at this point, and that 2 of them are possible or probable. Did I say probable?
2 of them are. That 2 of them are.
That 2 of them are possible or plausible, as they say.
Possible, plausible. Let's get back to the expert. Silke Stertz, I posed this question to her, what she thinks about the lab leak theory, that COVID-19 came from a lab, doesn't matter where, because of gain of function research?
There's no evidence whatsoever for a lab leak hypothesis. But on the other hand, there is a lot of evidence for a natural origin from an animal reservoir, from coronaviruses in bats. There is lots of evidence, experimental evidence, that supports that hypothesis. You can never prove. It will be very difficult to go back and find exactly the precursor. So this will remain open to some extent. But for me, as a scientist, I want to see data, I want to see evidence. And the evidence is clearly with the natural origin. The lab leak is speculation, and at least I have not seen evidence for it. So for me, this is a very clear case.
A lot of people online have shared, basically, it's a link to Wikipedia, and it's a Wikipedia list of the numerous biosafety incidents at laboratories around the world, not necessarily gain-of-function research related, but with things like West Nile virus. I'm just scrolling through the list here: SARS, Ebola, Marburg here in Germany was a kind of, it's not comparable today, but a kind of leak from a facility. Are you really convinced? Are you really certain that we don't run the risk of a low probability event, a leak from a gain of research lab with huge consequences, with huge risk?
We cannot exclude any risk at all. That is not possible. We have many different layers of safety. If something happens, another layer kicks in, but there is no such thing as absolute safety. We cannot exclude that. But, on the other hand, there is also the risk that a virus emerges from a natural origin causing a pandemic. And in my mind, that's a much bigger threat, and we want to prepare for that. So it's not that if we don't do it, we then don't have any risks. It's different risks, and we need to balance the benefits and the risks from both.
So if there are people out there worried about this kind of research, your message to them is?
We should be worried about the natural origin pandemic viruses. The risk is much higher that something happens and comes out of nature, as we've seen with SARS-CoV-2, than from a lab. And we need to understand these viruses to deal with them. It's not that we prevent any risk from not doing this type of research.
I guess the question I want to ask is, before we started talking here, you mentioned that you have a son, and Gabe and I have kids as well. And that really is what I think about when I think about these labs doing gain of function. Is something going to leak out? And is it going to create some sort of super pandemic that my kids are going to have to live through? They're going to have their lives upended. They might get infected. Who knows? Do you believe that this kind of research is making your son safer in the long run, as opposed to creating something that's going to be riskier for him?
Yes, definitely. Our lifestyles have changed so much. We have taken away the space from many wild animals, are in much closer contact to them. We travel worldwide, everywhere. So we are in closer contact with wild animals that have viruses, and we travel and spread things worldwide very quickly. These are ideal conditions for pandemic outbreaks. And there are loads and loads of viruses in nature. We need to figure out how to deal with that. That's our biggest worry, not what we do in the lab.
Just out of curiosity, another critique I see online are all these people saying, well, it's all the gain of function researchers who want the gain of function research to keep happening because that's their funding, that's their job, and if it stops, they have to do something else. Is there any truth to that at all, or is that just?
No, it's the entire field. And, no one of us questions that these types of experiments need to be tightly regulated and be subject to thorough checking and approval. No one objects to that. But often what people ask for is just ridiculous and would cause, so much bureaucracy for experiments that are really not problematic. It's this overregulation that a lot of people are worried about in the virology community, not just the ones that do gain of function. This is, I think, the biggest issue. No one questions that, these experiments need to be regulated, but is this going overboard? And there are lots of calls for that, to cancel anything that has remotely to do with gain of function, even if it's not gain of function of concern. That's what we're worried about.
Just out of curiosity, why don't you conduct gain-of-function research?
My focus is on how viruses enter their host cells. It's more a cell biology approach. And so currently I do not work with this topic of transmissibility or pathogenicity. And in those areas, these approaches are much more required. I work basically on a cell level, not on a whole animal or human level. So for the types of questions that I'm interested in, these are not relevant.
So when you look at how people are discussing gain-of-function research right now, you must just think people are nuts, right? Completely over-exaggerated hysteria.
I don't know if it's exaggerated. I think it's just the people that do not fully understand influenza virus biology and the research really in detail, that they are a lot more vocal than our virology community. I think the issue at the moment is that people are not speaking up. The experts are not involved enough to really explain to the public why one is doing what. It takes time and we're all super busy. Some people may also not be so comfortable with it, but I think this is at the moment the problem, that the other side is very vocal and the research community is not.
Is it kind of a thankless job to be Silke Stertz right now, sitting here talking to journalists, defending a kind of research that it feels like most people around the world kind of hate?
I don't think so. I'm funded by public money, and I think it's part of my job to explain to people what we're doing and why we're doing it, and to also help and explain. And so I see that as an important part of my job. I don't think it's pointless.
Do you get these questions? Do you get these questions a lot? Do people are they constantly hounding you, saying, "Silke, come on, we got to stop this gain-of-function research. These people are unnecessarily harming the world"? Do people approach you about this ever?
It's mostly journalists. I think the people that strongly oppose the work are usually in their own circles, and the researchers are in their own circle. I don't think there is enough discussion going on. It's usually via journalists that pick up both opinions and then try and educate people. But I think there's not too much discussion between the two groups. Both keep to themselves, which I guess is also not great.
Journalists picking up the discussion and trying to educate people, I suppose.
Well, here's to hoping that we have educated you with this interview, you people listening out there, that you got something from Silke Stertz and from us riffing on the idea of gain-of-function research, because here's some more riffing. We got Vivaldi. Before it was some dude playing the clarinet. This is a band of four guys. Three. A cello, a violin, and an accordion. Playing Vivaldi, one of the seasons. Didn't he write a bunch of music about seasons?
I think 4. Beautiful.
Yeah. Again, the perks of living in Bonn.
Well,
it's happening every day.
It's strange.
Every day.
I mean, to connect this to what we've been discussing, and I really mean it, it feels like the world is kind of waking, finally, truly waking fully up from the pandemic, at least at this point in time in this part of Germany. Everyone's out and about. Music is filling the air, even in a way that it didn't last year as much. It just didn't.
It's taken us a while.
We're back.
To recuperate.
Yeah, we're back to a point where we can look all the way back toward the beginning and try to kind of assess what we know, what we don't know, and how even a kind of experimentation or research that it very likely has nothing to do with the origin of this pandemic. But it could
But what might have something to do with preventing the next one.
Right, and so it's important still to talk about it now, even if it had nothing to do with this pandemic, because who knows? We're human beings, we make mistakes, and things get out of labs.
What are your guys' thoughts on this one? Not only with regard to where COVID came from, but also, should this research be done anywhere in the world? You think Silke's right? She's a human being too, right? She could be wrong.
She could be. SU@dw.com.
DW, made for minds.